Hi, everyone. Welcome to the session number 5 on computational analysis. I'm Gary Bader. I'll be chairing the session. This slide we've been showing throughout the meeting, showing the organization chart of the HCA, and this session is focusing on the analysis working group. One of the goals of today's meeting and the breakout sessions that we'll have will be to bring a lot of new ideas and energy into the analysis working group. And so we have invited some great speakers who will come on soon. Shila Ghazanfar, Lotfollahi, and Julio Saez-Rodriguez to motivate the interesting computational challenges that we all face and thinking about how to solve those challenges. And then we'll go into the breakout sessions where we will talk about how to energize the community, bring new people in, discuss the major computational challenges for the HCA and how to solve them, and also think about how to use all of the millions and eventually billions of data points that we're generating. So with that, I'd like to welcome Shila to the stage to start your talk.
Gary. Okay. Hello, everybody. Excellent. So thank you so much for the invitation. It's really, really wonderful to be here. So before I begin with my talk, I'd like to recognize and pay respect to the elders and communities, past, present, and emerging, of the lands that the University of Sydney's campuses stand on, the Gadigal people of the Eora Nation. For thousands of years, they have shared and exchanged knowledges across innumerable generations for the benefit of all. So my scientific mission is to use capacity and methodological creativity to solve emerging data problems in biomedical research. I'm a research leader in the Sydney Precision Data Science Centre, and I acknowledge funding from the Australian Research Council as well as the CZI. So what I'll talk to you today, I guess really is motivated by the recognition that biology is spatiotemporal, right? So if we consider, many different modes of biology components, we have like an innate axis of space in which these processes are occurring, but then these are dynamic that are happening across time, right? So we have emergence of cancer, we have response to a perturbation, for example, wound healing, and we have normal developmental processes as well. So in terms of the data sets that we have available to us, we've heard a lot of different talks over the last 2 days. Really, we can kind of think about where we're at the moment. We seem to have this sort of balance between the sort of single-cell transcriptomic techniques, where our ability to measure at scale is actually giving us a bit more ability to get to a dense time course in temporal. But then we're lacking in terms of the space, right? And so our current limitations in the cost and so on for spatial omics means that we're sort of at the stage where we have the spatial information, but perhaps a bit limited in terms of the density of time point characterization. And so ideally, we would like to have more of these. We would like to integrate all of our data together. In terms of the data sort of representation, we could characterize it sort of in terms of matrices here. So, if we think about SCR and ASIC, we have this breakdown of our cells by our genes. We may have some densely sampled time courses. And then, what I have grayed out here is actually, that spatial component of our cells, right? The space didn't go away. It's just the fact that we didn't measure it. And then, on the other hand, we have this sort of characterization of spatial omics data sets available to us at the moment, where we have, our cell gene plus a spatial component, but we may be a little bit limited in terms of those time components there. And so, I've been thinking about this problem of how to treat spatial coordinates in computational modeling. And I'm going to mention just one approach, and then I'm going to go deeper into another approach. So, one way of considering the spatial coordinates in computational modeling is to think about it as a data integration problem, right? So we have a set of derived features from our space, right? So we had a really wonderful discussion yesterday in one of the breakouts about how we could derive new types of features from niches, from neighborhoods, and so on. And so, if we want to actually integrate this with our single cell data, how would we go about doing that? And so this is the one that I'll just mention briefly. I've developed a technique called StabMap, Stabilized Mosaic Single Cell Integration, using unshared features. And the idea here is that we can utilize essentially the shared information between our multiple data sets to be able to sort of propagate information across multiple data sets, even ones that have no shared features at all. And so the assumption there is that those few shared features might have some information that allows us to propagate across. And so this was my final project from my postdoc with John Marini and a collaboration with Carolina Gabentif. Yeah, so I'm not going to talk about that one much more. I'm actually going to talk about another idea that is unpublished. So this is thinking about direct modeling, and I'll describe a method called WSIR, and soon you'll know what that means. But really, the way that we want to think about this is that our data coming from spatial omics, we can characterize it in terms of our X matrix, our explanatory variables, and then a Y, where Y is our outcomes or our responses. Right? And so, if we characterize our data this way, then it sort of naturally comes to be that, we may want to be thinking about how can we transform X so that we can best represent Y, right? So a method in statistics that deals with this problem is called sufficient dimension reduction. And so I'll just go back a slide just to note that this project is joint with my colleague at the School of Maths and Stats at USyd, Dr. Lynn Neem, and is led by Max Woolard, a PhD student, and Dr. Pratibha Panwa, a postdoc in my group. So sufficient dimension reduction essentially hinges on this idea that we can think of our X matrix. We want to be able to identify what is a reduction of X such that all of our information about Y is still preserved, right? So essentially, I want to ensure that if I know this reduction of X, then it's basically independent. Y and X are basically independent of each other, so long as we know what R is. And this approach is called sufficient dimension reduction. Now, if we assume something about linearity, right? So if we assume that this Rx is basically a linear combination of our X and a set of coefficients, then this becomes something called linear sufficient dimension reduction. And so what we really have as our model here is that our Y, which is spatial coordinate, for example, is equal to some function of linear combinations of our X, right? And so note that it's not F that's linear, it's actually the inputs to F, right? So this F function could be completely weird and wacky and nonlinear and everything exciting. So a technique for dealing with sufficient dimension reduction was really built from this paper from 1991 in statistics that set the scene for sliced inverse regression. And so I know there's a lot of formulae here, but essentially it hinges on the idea that to be able to estimate B, remember those were the linear combinations, we it's enough actually for us to know something about this lambda matrix, right? So we can perform eigendecomposition of this lambda matrix rather than our B matrix, which is a little bit harder to characterize. And so if I sort of zoom in on this lambda thing here, and we sort of pay attention to it, what we're saying is that this lambda is the covariance of the conditional expectation of x given y, right? So essentially, the way that we can estimate this matrix is by taking our data set, so our cell by gene and our y spatial information, we're going to rearrange our cell rows so that we actually have all the cells that belong to similar values of y together, right? And this actually gets us to that first bit, x given y. So for consistent values of y, I want to get x. Now, expectation for the statisticians in the room, that just means taking averages, right? So what we do is we then take averages along those small slices, and we get a slice by genes matrix. And then the other thing is taking covariance, right? So from that, we can calculate a covariance just as we would normally. So now a challenge here that we sort of saw was that if we treat spatial coordinates as these slices, we can sort of break them up into the slices, we can take averages, we can calculate covariance. One big challenge is that we actually lose the slice-slice relationships, right? And so this is the novel component to our work here, which is to incorporate the slice-slice similarity. And the way in which we do this is actually to not perform eigendecomposition of our XH only, but to incorporate this weighting scheme that takes into account the spatial relationships among the slices. So where does this leave us? So what that means is that we can take our spatial omics data set through the WSIR model. We first get out our WSIR directions, right? And so this is a genes by directions loadings matrix. So if you're familiar with PCA, this is it's essentially PCA. But then from that we get our low-dimensional embedding of our cells according to these directions. Now, given that this is a linear approach, any additional data set, you only need that X matrix to be able to project it. So that means that any single-cell RNA-seq data set can be projected into a space that is spatially aware via this sliced inverse regression setup. And so in the last sort of few minutes, I'll go through a very small toy example of a donut. And so in this simulation, we essentially have a lot of non-spatial variation, and then we have a little bit of spatial variation. So we kind of want to find that spatial needle in the haystack. And when we apply WSIR and our suite of other techniques, what we see is that the SIR type approaches, as well as linear discriminant analysis, are able to recapitulate that angle and radius of our points there. But then things like partial least squares, and in particular PCA, which I would say is our bread and butter of dimension reduction, completely fails here. And that makes sense. And so then, looking at distance correlation, we see that there's ability to recapitulate the spatial information just due to our dimension reduction in the first place. And so I'll just have one case study looking at 10x xenium breast cancer data set. So here we focus just on the stromal tissue. And I'll note that actually performing this dimension reduction on 40,000 cells took less than 1 second. This is because it's a linear approach. So what we do is we perform WSIR and we perform PCA. And if we plot these in space, what we can sort of see is that there is remarkable domainness in the WSIR approach. And this makes sense because it's a spatially aware approach. Now, if we perform clustering and we look at the breakdown of the different cell types, so this is a confusion matrix, we see that they're very discordant to each other. And in particular, this PCA cluster really has these two extra groups. And so if we focus on these two extra clusters, 2 and 4, so that's the red group over here and the green group over there, if we take those clusters and perform a differential expression analysis, we can see that some of the highest differential genes are those related to prostaglandin and aquaporin, which correspond to the emergence of tumor-infiltrating lymphocytes. So I just want to give a little vignette there of the ability to use a linear spatial approach to get something informative from what otherwise would have been like a homogeneous set of cells. And so, in terms of further considerations, I've shown that this is a sufficient dimension reduction technique, and it retains all of the information about the spatial response. It can handle multiple samples, and it essentially highlights features and patterns that are relevant to the spatial response. So it may miss cell type information if it's uniform across your tissue. And a key exciting thing to me is actually employing this in alternative coordinate systems or using different types of slice mechanisms. And with that, I'd like to acknowledge the team again and thank you for your attention. Thank you.
So we're a little bit short on time for questions, so we're going to go right to the next speaker. So Mo, I'd like to welcome you to the podium.
Okay, I think it's the last slide, so it should go to the first one. Yeah, thank you so much, everyone, and thanks for waiting. Okay, so I'm going to talk about generative learning of gene programs. So I started my group 5 months ago at Wellcome Center Institute, an associate faculty in the University of Cambridge. And my group works at the intersection of machine learning and biology. And today I'm going to talk about a few ongoing research in the lab about using machine learning to learn gene programs. Okay, so gene, I think you should see this slide. It's fixed there. It's only changing here. Should be this. Huh? Okay. Oh yeah, there you are. Okay. So you see my dots now. And okay, so I'm going to talk about gene programs. So gene programs are a set of genes that are involved in a function. So for example, genes that are activated after an inflammation cue, or genes regulating metabolomics, or genes that are up and down regulated after transcription factors. But we know that they exist, and then they get up and down regulated in response to perturbation and environment, and they cause shift in cell state or cell type, and we get a snapshot using single cell data of how these gene program changes. But these systems and the single cell data that we have, most of the data from HCA, they are in vivo data. In order to intervene on these gene programs and change the cell fate, we need to have an accurate in vitro system. But the in vitro system is not perfect, so these gene programs will be shifted a bit, so you can't really recapitulate all the cell state that you might get in vivo. So this started a question for us, and then I and Rosa sat down, because she's doing a lot of in vitro work, to see how can we capture these differences between in vivo and in vitro. And we hired a PhD student, Marie, working with a machine learning scientist in my lab, who are trying to address two questions. Which gene programs are differentially expressed between, or are different between in vivo and in vitro? Can we use this to go back in the lab and then optimize our in vitro system to be as close as possible to the in vivo system? Okay. So methods to learn gene programs are not new, so there are a few methods out there. So one that I developed in my PhD, there is a really good one from Donna Pierce's lab called Spectra. What they do is, instead of giving you a local embedding, something like this, where you don't know axes of variations, they'll give you interpretable embeddings using the domain knowledge about the gene programs, or they learn de novo gene programs from scratch. But the paradigm shift is that you go from these global embeddings to local embeddings that you know the axis of variation and you know the changes. So, but these methods, the interpretability comes with a price. So, for example, the one that I did is a VAE, and then you have to make it linearize and put a mask and a lot of other tricks to make it like interpretable, and that reduces the capacity. So from a highly nonlinear model, you go to something in between. And there is a scalability issue with some of these methods because they're factor models, and they might not scale well, but they give you interpretable embeddings, so these local embeddings. So can we go and then have a method that is capable enough, so like it has a high learning capacity, it is scalable, so you can train it on millions of cells, and at the same time have interpretability. So transformers, I think you've probably heard of like single cell foundation models and other type of foundation models. The backbone of these models are transformers. Transformers are high capacity learners, but they're not necessarily interpretable. So we try to have all of these things in a method that we call GP-former. And the GP-former, it basically eats your gene embeddings from your favorite foundation models, or you can actually train your own foundation models and feed your gene embedding into GP-former. And the GP-former learns a local representation per gene program. So instead of learning one scalar vector for each gene program similar to the factor models, now you learn an embedding per gene program. And you can contextualize your data into different gene programs. And then there is a second set of transformers that combine these gene programs into a global embedding to reconstruct the gene expression. So you have global representation of the data, and then the local representation of the data into gene programs. And there is a module that we call a GP Finder that kind of learns novel gene programs too. Okay. So after we developed the method, we went back to the first question. And Browser is interested in developing macrophage models, in vitro models, because they're important in immunity, and then with in vitro models, you can do screening. So the question was how accurate these macrophage in vitro models. And then two amazing postdocs in her lab, Roser and another Roser, and then Sung-Jung, they developed this, and then they generated this massive in vivo fetal immune data, which can provide an in vivo atlas by curating few existing data set and adding fetal bone marrow. And so they complemented this into this atlas. And then for us, the question was, can we integrate these two data sets and see how accurate our in vitro model is? And if you use any integration methods like SCVI and then our GP-former, but global embedding-based models, you'll see that things fly in the scene, and then you UMaps and nothing integrates. So you could have taken two directions. Go and try the next integration methods to push things on the top of each other, but they're kind of like apple and orange, so you shouldn't really do that. And so the question was, can we move from these global embeddings into local embeddings, because we know there are some similarities and some differences, and see whether it could give us some insight about the differences and similarities, but in the gene program space. Okay. Now we switch to gene program space, because using this method, you can get an embedding from the gene programs too. So what Marie did, the PhD student, she used the embeddings that you get for cells to rank the gene programs that are different using Washington distance between the in vivo and in vitro. And what you see are the gene program ranked from like left to right. The top one, which is hypoxia, is the most different gene program. So now you can project the data into gene program embeddings. So you see in the hypoxia gene program, things are different, because these cells that are the cells that were in vivo and in fetal development, they were in hypoxia state, whereas the in vitro system, it was not in that state. So this could give you a clue that how can I make these cells to be more hypoxic. Another one is SOX4. So SOX4 have been reported to be utilized in early development of macrophages. And in that gene program, things are kind of aligned. So that could show you that maybe this gene program or this in vitro system captures the early state of macrophage development. But then there are other gene programs here. So we went deeper into one of them. So it was MAF. So MAF is known to be involved in macrophage development and then differentiation, and also leading to terminal stages of macrophages. So MAF was among those top lists, but then Marie went to the gene expression space to see the downstream target of MAF. And you can see, if you compare that between in vivo and in vitro, the genes are downregulated. So that could be a clue that these markers and then obviously the macrophages that you get in vitro might not be fully differentiated. And then we looked at these downstream targets in the in vivo data, and you can see in the dot plot there, from early stages of development toward the end stage, you can see the markers actually increase. So once you go and then you go further in development, the maturation gets higher. So if you go back and look at this data in comparing in vitro macrophages and in vivo, you can see those terminal stage markers of macrophages are not there. So using this type of analysis, you could see the gene programs that are differential between in vivo and in vitro systems, specifically when you combine systems that might not be fully integrated using data integration methods. And then what we're doing now is, like in validation stage, going back to the lab and see if we can intervene here and then have a better differentiated macrophage. OK, so the second part of my talk is about, once we learn good gene programs, can we intervene on them? Because with single-cell data, you might only get a snapshot of the data. So if you also do a time sampling, you might get 1 or 2 samples, but we know that the gene programs change continuously. So there are gaps here that you might not be able to experimentally capture. And so the question is, can generative AI help us here to fill the gap and see if we can predict how the cell change and how gene programs change in response to perturbation? So there is a really strong body of work from the community. So I developed the first model called SCGen, so the single-cell generator, to predict the response to perturbation. And there are I don't know, 20, 30 methods out there now using optimal transport. And the latest methods are based on newest generation of diffusion models or flow matching. But there is an issue that these flow matching models, as opposed to language and vision, we don't have good embedders of single-cell data. So they operate on PCA space. So these methods that generate cells, they have no clue about the gene. The only thing that they get is an embedding. Whereas we're interested to basically operate on the gene level, so the model knows about gene-gene interactions from scratch. OK. So this motivated us to develop a model that we call CellGen. This is a collaboration with Gosha Trinka, led by a PhD student, Kevin Lee, and then machine learning scientist in the lab, Amir. And so we formulated the problem, the generative problem, to work on a sequence similar to language translation. Can you look at the unperturbed state, or a healthy cell state, and look at it as a language 1, and then you learn a translation between language 1 and language 2, which is your perturbed state, or perturbed state in time. So you can see here, that is how neural translation works. They are seek-to-seek models. So you can see you feed in a sequence of English word that is good, and it will give you a German version of it that is good. So it's an encoder-decoder architecture. But these models are generative. So once you train them, as you do with ChatGPT, you can write the prompt, and they will generate more text. And then they have been applied in other settings. Like, for example, this is a sketch from ESM3 model, where you provide a model with a backbone of GFP protein, and the model iteratively generates a GFP-like protein. Can we do that with cells? If we model this and then throw in a lot of single-cell data, can we have sequence-to-sequence models? So we worked together with Gosha. So she works on CD4 T cells, and then she's interested to see whether you can activate them and understand how the gene expression dynamic changes in CD4 T cells, and can we predict them? So we trained the model on the CD4T dataset from Gosha's lab. And this is the early stages of generation. So similar to the backbone of the GFP, the model kind of understands there might be 2 cell types, CD4 and then CD4 memory and CD4 naive, but the substate of T cells are kind of blurred. They're all over the place. But when you iteratively generate data, they kind of shape up. So you can see that even at the last iteration of the generation, because it's kind of demasking. You mask genes and you add genes, and toward the end of the stage, it can predict or basically generate counts and sequence level information of T-cell substates. Okay. So now we worked with, and that one is still in the validation stage, but that's like one cell type. Can we formulate and model complex systems? Here is another example that we worked together with Most Hanifa and then postdoc in her lab and my group members to see if we can generate generate the data which is more complex, that there is response to a perturbation, in this case, perturbation LPS. So LPS binds to CD4 and TL4, toll-like receptor 4, and then it activates and kind of causes cells to secrete cytokines. So you have like B cell changing, you have monocyte changing, T cell changing. So it's much more complex system compared to the CD4 T cell. So we generated the data set from a bunch of donors, and then we have normal cells, and we activate the cells using LPS, and then we measure two time points. So the first one is 90 minutes, the second one is, if I can see it, like 10 hours. And then we ask, so if you look at the data, so you can see we annotated cell types, and then you can see the original data, you can see the shift between the LPS, and then we also quantify there's a shift in also cell composition and also expression, so we have both of them. So then what we ask is, can we predict what happened in between? Can we predict 6 hours? So the model has not seen, well, it has seen a lot of PBMC data, it has been trained on a lot of data, but it has never seen LPS before this fine-tuning, and then never anything between 90 minutes and 10 hours. So we asked the model, predict the perturbation response at 6 hours. And then the model generates these sequences, and you go back to the expression space, and this is what you get. I asked you to kind of appreciate here is like, it's not an interpolation problem. We're operating in a high-dimensional gene expression space. So all of these cells have a nonlinear response. So that's why you have like only monocyte changing. So it's a very OOD problem in a case. So, and you can see that the model kind of recaptures global cell types, so like large cell types, but also captures rare cell types, even hematopoietic stem cells, and it can recover those. But then, so this was the validation experiment, and then we compared it with the data that we generated in the lab just to see whether it matches. It didn't just change the 10-hour or like 90 minutes, and then the closest similarity based on Washington distance is 6 hours that we validated. Okay. So with this, I'd like to summarize. I think, so we'll release these models. The first model would allow you to learn gene programs and also query them at scale. So trained on HCA data, I think it would be super cool that we can project the data and then look at all the cell types across different programs, and we'll provide you a dictionary of gene programs in this way. And with the second one, which I'm super excited, for the next wave of human cell atlas data, where we have healthy now, but now adding disease and perturbation, there will be a lot of gaps. No matter how much money we put into this, there will be a lot of gaps in this space, especially for perturbation. So I think it's a very interesting direction to push this generative AI side of modeling, but also generate data to train these models, which I think is very important. Otherwise, I think Dana sometimes calls it garbage in, garbage out, so this will happen. But I think this could be one of the interesting things in the human cell atlas discussion to discuss. Okay, with this, I'd like to conclude, and then thanks to funders and students. And if these things are interesting, please reach out. And then I'm looking for good students, postdocs, if you want to leave your lab and join us. Yeah.
Okay, thank you very much, Mo. And we'll invite Julio next. We'll just take questions at the break because we're running a little bit late overall.
Yeah, hello everyone. It's great to be here and be able to share with you some of our thoughts. What I'd like to talk about, in a way complementing also what we just heard, is how we can leverage knowledge, so the things that we know about in analysis of atlases to better understand gene and multicellular programs. So the idea is there is a lot of knowledge out there, in part in the context of the atlases, but also molecular knowledge that is curated in different databases, such as the target genes of a pathway or which ligands bind to which receptor. And by using this knowledge, we can use it to, from our data, on one hand, extract number of features, and by doing so, reduce the dimensionality. So rather than 20,000 genes, maybe a few dozens of pathways. And also, these features are close and built on relatively well understood biochemical processes, so they get us a bit closer to the mechanism and potentially towards true causal mechanisms, although it's, of course, a much more broader and complicated question. So we just heard before in the discussion in the panel around where AI is going to help us, and also Aviv in her talk yesterday alluded to this. And so part of the process of building all this knowledge for the analysis is really the curation and integration of existing knowledge. And that's quite a tedious job, so you could think, well, now we have a large language model, so we could use them to help us to build all this knowledge together. And however, if you ask state-of-the-art model, ChatGPT-4, to draw for you a model of the EGFR pathway, arguably the best understood pathway, signaling pathway in humans, it will give you an output like this, which maybe from the distance looks like it makes sense, but if you look closer, it's completely useless. And so we were talking actually yesterday with Gary about this, and he was also saying how these models, almost by design, will always hallucinate, so we need to come up with ways to harness their power, but also control it. And that's why we and others are interested in building open source platforms for applying them in the context of biomedical research, and in particular, to use this knowledge, the things that we trust, to ground the LLMs and by leveraging via knowledge graphs. So still we need to curate knowledge for use it in the context of serious science. And we have, over the years, been building a resource called OmniPath. This is also in collaboration with Tomas Coxamaros, who is here in this meeting, where we try to bring different resources focusing on sign-directed knowledge about protein interactions. And this complements things like Pathway Commons that Gary has been also co-leading, and other things like String or other efforts to bring together knowledge. And the reason why you want to bring different resources is because they have different focus, maybe more immune system, or maybe more oncology. Maybe they are only really things that are really highly curated, but narrow in focus, or maybe they are more lenient and they bring different things. And then by having these resources, you would like to know what's the right one for your analysis. So the same way here with analogy with the glasses, you like to know the right lens that is best for you to see. You like to find the right resource. No? And that's the question then. So which resource should I use, together with which method, for what? To answer which type of questions. And to answer these three questions here is we need to benchmark both the resources and the methods, which is something also that I like us to think about today, also as we move into the breakout sessions. And so as an example, if we have single cell or spatial transcriptomic data, I can use knowledge about target genes of the pathways or target genes of transcription factors to estimate their activity in different cells, cell types, and so forth. And we and others spend time looking at this, benchmarking it, and seeing if they're robust, if they work on single-cell data. And to facilitate this, we also built a small toolkit called Decoupler, which is part of SCVerse, that provides you different enrichment methods, from overrepresentation analysis to gene-set enrichment analysis, to the types of analysis in a streamlined manner. And just to illustrate the value of this relatively simple analysis, so this is work with Lucas Schirmer working at multiple sclerosis patients. So this is a classic Visium slide. If you look on the left, we were interested in a particular transcription factor here, RFX5. If you try to look at it, because of how sparsely measured it is, you don't really get a clear pattern of what's happening. But now, if instead you look at the target genes, so the genes that are controlled by RFX5, and you just enrich for them to get an estimation of the activity of the transcription factor, you get a pattern that makes much more sense and that really follows what's known of the biology of a particular inflammatory region in the brain of multiple sclerosis patients. So these are simple ways where we take the existing knowledge and we use them to extract features that are helpful for us. But of course, we can go beyond using the knowledge as it is, and we can try to contextualize it with single-cell data. And we just heard from Mo about XPMAP and the newer models, so there are others like Spectra, Movie, and so forth, that allow us to take the knowledge and adapt it and contextualize it to specific cell types or specific conditions. And so we took a bit of a deeper dive in the specific models that uses single-cell RNA and ATAC to estimate gene regulatory networks. So these methods have exploded over the last years with the advent of single-cell attack data. And all the methods work differently, but they all use this data together with information about transcription factor motifs. And you could then ask yourself, OK, there are many methods. Which ones should I use? Now, you can take any data set. This is a simple PBMC data set, and run some of the state-of-the-art methods listed here, and also some of the pure knowledge-based methods. And you will see that you will get quite different results in terms of the edges that define, or even the target genes that they identify as in a particular context. So then again, this calls that we need to benchmark these methods. And in a work that we are now trying to wrap up, led by Pau Badia, we've been trying to see how well these methods are able to, let's say, predict the new experiments or recapitulate existing knowledge about activity of transcription factor in different contexts. It's important to benchmark methods to understand what are best for and how to use them. Another context where this is important is cell-cell communication. Now, by having single-cell and spatial data, we cannot just look what happens in the cell, but how they interact. And here there's been a lot of great methods, like also CellPhoneDB from Sarah's lab. And to get a bit of an overview of these different methodologies, we built this framework called Liana, starting first with a method for dissociated single-cell data. And then when we again benchmark them in different ways, in the development of the tool, but also in the context of the open problems framework developed by Malte, we found that they give you quite different results, and they seem all to be getting some signal, but it's also not so clear what they are best for. No? So again, there is this need to understand and benchmark the methodologies in this space. This is from dissociated data, but now as we have more and more spatially resolved data, we can bring different methodologies, including some of the things that Sheila talked about before, to study cell-cell communication in space and try to link so that with even multimodal data and intercellular knowledge to start to better understand communication between cells and inside the cells. But I emphasize that we need to benchmark all these approaches. And most of these methods were originally developed to look at specific experiment or comparison of conditions. But of course, in the context of looking at atlases, we like to look across many different subjects and try to identify also in an unbiased way patterns of cell communication and other processes. And in our case, we work mostly in the context of human disease. So we're interested to understand how different cells are coordinated in response to disease, as well as different pathways, as we discussed before, and changes in different states in the development of specific disease. And to do this type of analysis, we have found quite helpful to repurpose MOFA, the framework from Oli, Stekle, John Marion, Wolfgang Huber, developed for multi-omic analysis, to repurpose it to look at multicellular programs. It is similar in spirit to, let's say, Dialog, developed by Shila for SCITD. And it allows you to find this axis of variation across patient data sets in an unbiased manner. So Rico has a poster describing this approach. That then, once we have those axes, we can connect them to clinical descriptors, but also map them back on other data modalities, such as bulk or spatial or pathological data. And we've been doing this across different organs and disease listed here, in the kidney, together with the KMPAP community, Crohn's disease, lupus, and so forth. Just I'll tell you two small insights to the work on the heart. So this is work led by Jan Lancer, who is also here with a poster. So Jan is an MD, and we're together with Rico to reanalyze existing data of different heart etiologies. And then when you analyze all of them together, you find two main multicellular programs that seems to distinguish between failing and non-failing hearts. These programs capture both processes that are happening across all the cell types involved, like response to stress or inflammation, but also cell type-specific processes, such as a metabolic switch in cardiomyocytes or expression of extracellular matrix in fibroblasts. Then these features, these multicellular programs, we can, as I mentioned before, project them in larger bulk data sets. So earlier on, we had collected over 1,000 bulk transcriptomic data sets. And if now you take these multicellular program signatures and you project them back on those, you see that they can, basically, the multicellular programs are conserved, so they are largely enriched in the heart failing patients. But also you can use them to predict the potential outcome of interventions. In this case, we took data published last year where they look at patients that have had a device implantation in the heart when they are really in advanced heart failure stage. And then the signature that we found before of heart failure is able to predict if patients are going to respond positively or not to this intervention. So there's one example, but there are many others. So I invite you to talk to Jan and Rico, and also to say that the same thing can be done across organs. So Leonie has a talk tomorrow and a poster out there discussing how you can use these approaches to look at fibrotic processes across different organs, the heart, the lung, the kidney, and hopefully soon also in the liver. And with that, I'd like to finish. I'd like to thank the people in the lab, our collaborators, our funders. We're also looking for new people to join. We just moved to BI of Cambridge, so to do this type of work. And in the last seconds, I'd like again to emphasize what I tried to convey today. So we think there is a lot of value on leveraging knowledge to extract mechanisms from atlases and to better understand multicellular programs. But also, I think there is a big need to benchmark different methodologies. I alluded to this in the context of gene regulation, cell-cell communication, but becomes only, I think, even more important with the advent of foundation models that are so complicated and so powerful. We need to think really hard how to best assess them. Also, Mo organized earlier this year a workshop trying to discuss this. And I think here as a community, leveraging efforts like Open Problems, but also I'm part of so-called Dream Challenges, another community trying to crowdsource and benchmark problems. I think we can try to move forward to solve these questions. Yeah, thank you for listening, and happy to talk to you anytime.
Thank you very much, Julio. I think in the interest of getting time and getting the breakout sessions, I'd just like to thank all the speakers, Sheila, Mo, and Julio one more time, and then we can move to the breakout sessions. Is it possible to bring up the sessions? Okay, great. So the community session will be here. Thinking about major computational challenges will be on the 6th floor in room 1, and figuring out how to use all of the data we're generating effectively in all the different ways we want to use it in breakout session 2. So we can move to the breakout sessions, and yeah. Thank you.